So our second last talk of the session will be from Zhaoyuan Gong. Dr. Gong is a research scientist at the National Institute of Aging. We're going to hear about his work on exploring the link between myelin integrity and cognitive decline in aging, a comprehensive longitudinal MRI study.
Can you see the slide? Can you hear me?
Yes.
Oh, great. Okay, I'll get started. Okay. Good afternoon, everyone. I'm delighted to be here with you all. From the colleagues this morning and early this afternoon, we learned a lot about the validation of novel and existing qMRI techniques. Building on the theme of validation, I'll be taking us down to a path that showcases another dimension of validation, I would say. Specifically, I want to show you the validation of the usefulness of qMRI measurements. My presentation will focus on uncovering how myelin integrity, as measured by myelin water fraction, correlates with cognitive decline during normal aging. This talk aims to validate qMRI's relevance to aging process. So, globally, we are living longer than ever, a trend that underscores the urgency of understanding aging. Life expectancy has soared from 46.5 to 71.7. This rise is a marker of global health achievement, yet it brings the need to enhance the quality of life as we age. As the longevity gap closes, the aging society emerges as a crucial area for global research efforts, especially in cognitive health. That's because as we age, our cognitive faculties undergo significant changes. This graph captures the trajectory of cognitive decline. Fluid intelligence, which includes problem solving and processing speed and working memories, typically diminishes as we move through our 30s and beyond. Conversely, crystallized intelligence, representing our accumulated knowledge and vocabulary, remains stable or may even improve with age, as I'm expecting for everyone at this conference. This trend raises an intriguing question. How might qMRI metrics like myelin water fraction correlate with this decline in fluid intelligence? MRI is becoming an indispensable tool in aging studies, offering insight across various biological dimensions. For example, conventional structural MRI reveals ventricle enlargement due to atrophy and fluid dynamic changes during aging, while musculoskeletal imaging shows how TA muscles atrophy and fat penetration both are hallmark of aging. Cardiovascular assessment through MRI images can measure the arterial decline elasticity, a risk factor for the heart disease. Beyond those structure things we can measure with MRI, such as functional MRI studies highlight diminished connectivity in brain networks, particularly the default network, with implication for cognitive decline. Metabolic changes in brain are evaluated through MRIs, and that's showing distinct patterns across different regions and ages. Finally, as a shameless plug here, a paper from our group shows significant differences in myelin content between young and older subjects. So a closer look into this paper. It illustrates that quantitative measurements, such as an NMR-based matrix like T1, T2, magnetization transfer ratio, or biophysical modeling, such as myelin water fraction here, can effectively delineate normal aging process. Moreover, this diminished myelin content in individuals with mild cognitive impairment and Alzheimer and vascular dementia is captured in this image as well, highlighting myelination's potential as an imaging biomarker for cognitive disorders. As you heard a lot early, in vivo myelin mapping allows us to model the brain's microstructure, distinguishing between extracellular water and myelin water. Since Metcalfe's group introduced the first myelin water fraction map back in 1994, the field has rapidly evolved. Shanionis brought us this McDespot protocol in 2008, followed by Grace in 2012, and Vesta in 2013. Numerous other techniques and mechanism has been used to probe myelin content, and emerging new techniques, as you heard this morning, such as inhomogeneous magnetization transfer, water gap mapping, and cross-polarization between phosphor and proton, I do apologize, I couldn't cover all of them here. So research affirms the strong link between myelin content and cognitive function. In young children, greater myelin volume fraction in specific regions correlates with faster processing speed. Adults with MS exhibited clear association between myelin damage and cognitive impairment. Longitudinally, all recent paper found lower myelin content predicts more rapid decline in executive function, verbal fluency among cognitive normal individuals. Given the myelin's critical role in facilitating rapid neurotransmission, it is highly possible that it will longitudinally correlate with processing speed. Also, processing speed is a foundational element underpinning broader cognitive abilities, such as executive function. Building on existing research, our hypothesis is that myelin content will show a correlation with changes in processing speed over time. So in our study, we recruited 121 cognitive normal participants. They are taken from 2 ongoing aging studies. We acquired whole brain mapping of myelin water fraction using our BMC McDespot protocol, which you will hear more in later talk. The processing speed was evaluated through the digital symbol substitution test. And because of the concern of the specificity of this test, we also adopt a composite processing speed score, which is evaluated through digital symbol substitution test and trail making test part A. On the right, you can see the visualization of the longitudinal decline of those 2 processing speed measurements. We conducted analysis voxel-wise and across 6 white matter regions using linear mixed effect models. We're particularly interested in the MRI term, representing myelin content, the time term, representing the lapse between cognition measures and MRI scan, and most importantly, the interaction term between myelin content and time. So this figure of the regression coefficient maps illustrate the effects of the interaction term, the myelin water fraction term, and the years from MRI term incorporated in the linear mixed effect model I just showed you. The figure here only includes statistically significant voxels that is determined by using an uncorrected threshold of p-value less than 0.01 and a minimum cluster size of over 400 voxels. Quite stringent criteria. Well, close to no significant cluster were observed for the myelin water fraction term across all brain regions, as you can see here in the middle row. The longitudinal term, the years from MRI term, exhibited notable and consistent negative correlation with both processing speed and processing speed composite, as you can see at the bottom of the figure. More importantly, the interaction term revealed a pattern of numerous significant positively correlated cluster within several white matter regions for both processing speed and processing speed composite. This positive correlation means that a higher myelin content in those white matter regions effectively predicts a slower longitudinal processing speed decline. We'll see more detail in next slide. Okay, let's move. So zooming into regional details, you can see how myelin water fraction levels impact cognitive performance over time. The red, blue, and green lines representing low, medium, and high myelin water fraction values. There is a notable trend where lower myelin water fraction, which is the red line, corresponding to faster decline in processing speed across all brain regions. This relationship is statistically significant in all regions of interest for processing speed on the left, and for processing speed composite measure, except the occipital and temporal lobe. It is not surprising all those invested brain regions could collaborate and creating a cohesive network that enables a nimble and adept processing of data. Consequently, the detected association between longitudinal changes in processing speed and regional white matter myelin integrity align with this paradigm. So to conclude, our study highlights that MRI mapping of cerebral myelin content is a promising method for identifying potential imaging biomarker of cognitive health. Our result demonstrated a robust relationship between myelin content in brain and cognitive performance, particularly during normal aging. Specifically, a higher content of cerebral myelin is linked to better performance better maintenance of processing speed over time, indicating its protective role against age-related cognitive decline. So ongoing research will further validate myelin mapping as a sensitive assessment for cognitive health. With upcoming collaboration we're conducting right now, we will focus on Alzheimer and dementia population in the future. With that, I thank all the collaborators and agencies, and thank you for your attention.
Awesome. Thank you so much. Do we have any questions? Maybe one or two.
I don't think we have any, but I could start with one. I'm not sure if I maybe missed that, but I was wondering, like, when you show your plots, like you were showing time to MRI, and I was wondering if this metric was related to age.
Yes. So age is actually a separate. Let me go back to the statistical model if I can. Oh, here we go. Oh, sorry, too much. So age is actually here. So that's the age. So that's the age where the MRI scan is conducted, and the time for MRI is actually is the time from, because the cognitive measurement is over the time, so it's changing. But the MRI was only measured at one time point. So that times for MRI essentially just when you measure the cognitive performance minus when you measure the MRI. So it's really a longitudinal term over the time.
Okay, thank you for the clarification. We have another question from Margarita Lopez. What are the cognitive variables that you measure, and which battery do you use?
So for this particular study, we measure processing speed by digital symbol substitution test here. And because by reading the literature, there is a concern of this DSST test is not very specific to processing speed. It also could underline your executive function ability, as we actually see from previous study. Myelin integrity is correlated with that. So we adopted this composite score by averaging DSST and trail-making test part A, which in literature seems like that would be more specific to the ability of processing speed.
Great, thank you. I think we're going to move on to the next speaker.
Sure, thank you.
Thank you again.